Okay. So my background is analytical chemistry, and I've been studying environmental pollution for about the last 25, 27 years. But today we're going to talk about some plastic. And how do I... No, that won't work. Oh. Okay. So, since the emergence of mass-produced plastics around the 1950s, our appetite for these materials is increasing year after year after year. So right now we're currently making about 550 million metric tons of plastic every year. And what I find astonishing is that we continue down this route of business as usual, by 2050 we'll be making over 1 billion pounds of metric tons of plastic every year. So it's just a staggering amount of material. So anytime you give a talk about plastic pollution and microplastics, by law, we have to show this graph. But last year, I actually sat down and looked at it a different way. And one of the things I wanted to look at is how much plastic waste do we produce per person per day in the United States. And I found something interesting, that from about the 1960s to about 2000, we saw about a 40-fold increase in this number. But luckily, it looks like it's starting to stabilize right around maybe 0.25 to 0.27 kilograms per person per day. So it's a good thing that it looks like this number is stabilizing, but I still think it's too high. I think we really need to start lowering this number, and I think that's going to solve a lot of our problems. So anyways, anytime you make so much of any material, you have to ask yourself, well, where's it going? And the vast majority of plastic is destined for landfill, or it becomes fugitive of the environment, about 80%. Only a small percentage is recycled, about 12%, which has got a host of problems incinerating plastics. But also a small portion is only recycled, about 8 to 9%. And even that's kind of a misnomer, because what we used to do is just ship it off to some foreign country and calling that recycling. But that's starting to change. But anyhow, as a result of this, now it should be no surprise that we're finding plastic everywhere we look. Surface water has been incredibly studied, but also sediments and soil, which is a major sink for a lot of these materials. Indoor air is a major route of exposure to humans. In fact, look around, look at the floor, look at the clothing that we're wearing. I mean, we're surrounded by textiles, so it should be no surprise that indoor air is a major source. Food and beverages, cosmetics. I did a study last year where I was looking at microplastics and cosmetics, and I found that cosmetics are mostly plastic. In fact, I got some on my shoe, and I still can't get it off. I mean, it's incredible. I don't know how people get makeup off. But anyhow, wastewater, we know, is a major source of contaminant, of microplastics and plastic pollution. Wildlife, we've all seen, pictures of whales opened up and birds. But the other thing is, I was part of a team that was actually the first to discover microplastics in karst groundwaters. And it makes sense, right? Karst groundwaters are very open, porous systems, but they're very important because they account for about 25% of the world's drinking water supplies. So that was something that I got to take part of, and I think is relevant to today's conversations. So we know that there's exposure, but the thing that we don't know is there negative impacts from that. So we don't know what the human health effects are from exposure to microplastics, and we certainly don't know if there are negative ecosystem impacts from the presence of these things, but we know they're there. But there is things that we do know. And the first thing that we know is that persistence. These materials are designed to live forever. If you think about plastic bags or cigarette butts, anywhere from 5 to 20 years to degrade. And to be honest with you, in the world of persistent pollutants, that's a pretty long time. But now, let's talk about our plastic ring holders, our plastic bottles, our disposable diapers, up to 450 years to degrade. So a child typically uses about 7,000 diapers until they're potty trained. Those diapers will be around more than 5.5 times that child's lifespan. So I think just the sheer fact that these things live for so long is concerning. It's crazy, right? These materials have been designed to live forever, but many of them are only intended to be used for a couple of minutes and thrown away. The other thing that we know is that we add a lot of stuff to plastic. People think plastic is really clean, but there's a lot of stuff in plastic. In fact, a recent study tried to catalog all the additives used by the plastic manufacturing industry. And what they found is there's over 10,000 different chemicals added. They also took it a step further, and they looked at how many of these chemicals were carcinogenic, were endocrine disrupting, or known to be toxic. And they found that 25% of the additives that we use in plastics are known to be problematic. So I think it's more than just plastic. It's also the additives. It's also what sticks to these things. They act like little sponges, and when they get in the environment, they like to soak up any contaminants that surrounded them. In fact, many of the passive samplers we use in water quality monitoring are plastic materials. The other thing that we know is they absorb biological materials. And this is these images of microplastics here from a study I've done in Lake Muskegon, Michigan, Holler, Michigan. And what we found is right away, within 3 months, we accumulated a biofilm around these plastics. And when we look at the diversity of microbes associated with plastic, we find it's a lot different than the surrounding environment. So the diversity of microbes is different than on the plastic than what we find in the soil or in the surrounding water. And the other thing that I found is that biofilm also enhances its ability to absorb pollutants from the environment. In fact, for PFAS, if I take these same plastics, and I did this experiment, I wrote a paper about this, but I take these same plastics and I put them in the lab and I expose them to PFAS. They'll only soak up about maybe 25, 30% most. But as soon as they accumulate that biofilm, they start to soak up PFAS hundreds of times the background concentration. So that really enhances their ability to absorb these things. So these are all things that we know for sure. Now, you also hear all the time that there's no standard method for the analysis of microplastics. And I disagree. Actually, in 2015, the National Oceanic Atmospheric Administration, right? Well, thank you. I always get it mixed up. NOAA, they actually produced a method for the analysis of microplastics. And we were just gearing up our laboratory to do this work, and we took a look at this, and we actually thought this was a pretty good start. We thought that this had all the stages needed to be to really do this, and to do it in such a way that many labs can recreate it and do it in their own laboratories, because all you really needed was some glassware and some reagents and a microscope. But we did find that there were some things that we can improve. So the way it worked back then is what you do is you'd first filter your water sample. The sieve size was 0.3 millimeters. Then you would digest it to remove the organic matter, and then you would separate it from the inorganic matter by just putting it in a salt solution. Then you could throw it on a filter and look at it under a microscope. So we looked at this and we thought, this is pretty good, but we could probably really improve this. And then one of the first things that we wanted to look at was that size. We didn't really like the fact that it was 0.3 millimeter. We didn't think that that was low enough. So keep that number in your head, too, 0.3 millimeter. We're going to look at it later. But anyways, the other thing, too, is we didn't like the density part, the density separation, we called it, because they were using table salt solution. And that only gets you a density of about maybe 1.2, maybe 1.3. But if you do that, you're going to be missing a lot of plastics, because PVC and there's other Teflon's really heavy plastic, but a lot of those plastics would be missed by that method. So that was something else that we looked at. So we played around with it, and we think that we can improve this method. I think this was published back in 2020. But we actually fabricated our own sieves to do smaller sizes. We're able to get down now to about 20 by 20 micron in size. We played around with salt solutions for a year, and we found that potassium iodine was a great salt to float our microplastics in. We actually fabricated our own density separators. I got a picture of that there. But that's kind of the working method we had back then. So we had this cool method. We started to play around with real samples. And what we discovered right away, that there's another major flaw with the way people were doing things. So the way it works right now, or not right now, but back then, is when you were looking at microplastics, you were just counting particles. And people were reporting this thing, the number of particles per liter. And it's kind of a meaningless unit. And the other thing, too, is you can have a particle that's, a fiber that's maybe 500 microns in length, but after we're done taking it back to the lab and doing all of our magic to it, you might break it into 10 particles that are 50 microns in length. So this number, this particles per liter number it drove us crazy. So what we started to do is we started to measure the size of every single particle we would find in a sample. And then what we discovered is we could actually do something kind of interesting, is we can actually estimate the mass. So what we did is we determined what's called the ferrite minimum maximum, which is the minimum and maximum diameters, and then we kind of drew an ellipse around every particle, and then we were able to estimate the volume. And then from there, if we knew the identity or we made some assumptions, we can address the density, and then we can actually calculate the mass. And that makes a big difference. So for starters, I'm going to show you some data from up in the suburbs of Chicago. There's these data graphs that I'm showing you are exactly the same sites, but the difference is how we report the data. So the graph on the left is showing you particles per liter. And if you looked at this, you would say, oh, that second sample is the highest in microplastics. But when we look at it in terms of estimated mass, that first sample is actually the highest, okay? So really, it's the question what you're asking, you know, which sample has more microplastics in it? And it's all related to size. So, but this micrograms per liter, this is what we typically use in the environmental field for reporting contaminants. This is a number that we're familiar with. This is something we can work with. The modelers can work with it. The toxicologists can figure out, you know, reference doses and concentrations for EC50s, and all that stuff can be done with micrograms per liter. So that's why we think that's so important. The other thing that we noticed right away, size matters. So if you look at these graphs, you can see we're going from the smallest size to the largest size, from left to right. And what you'll notice is that as you move down in size, the occurrence drastically increases. So these two plots are this is what we see almost across every environmental compartment we look at. So I have theories of why these two are different, and if you want to know why, you can buy me a beer tonight, and I might tell you. But anyhow, we'll move on. But the point is that the size is extremely important because as these things get smaller, they have a higher potential to cross biological membranes. They are more mobile in the environment. They're much more difficult to treat. And they're much more likely to get in a well, right? The smaller the size. So that's why it's so important. And then when we started to see this data, the next thing we said is the smallest measurement we can do here in this data was about 400 micrometers squared. So we thought to ourselves, what would happen if we could see down even lower? Would it be even higher? So that's when we started to work on nanoplastics. So the way we started is we had a great workflow for microplastics. And in the beginning, I told you I filter that sample, that water sample. So everything that's on the top is greater than 5 microns, so we can backflush that and process it for microplastics. But then what we started to do is we started to save that stuff that came out of the bottom of that filter. We called it the raffinate. And that's where our nanoplastics are going to be. So then that's the portion that we started to play around with. But one of the problems was the technique that we're going to use to measure the nanoplastics. We need to get it down to about 20 microliters. And that's not my blue nail polish. Blue's not my color. But my colleague, Sophie, is showing you the capillary tube this is how we introduce it into the mass spectrometer. But the problem is typically our samples are usually 4 liters or 10 liters. We actually try to get as much large a sample as we can. In fact, we've worked with drums now, size of 55-gallon drum sizes of samples sometimes. But anyways, we needed to get it down to 20 microliters. So what I did is I built this plastic-free stainless steel ultrafiltration membrane system. And I found a membrane that's got a molecular weight cutoff of about 1,000 Daltons, which I'm figuring is about right around 100 nanometers. And what I could do is I could pump that sample through that membrane and basically reduce the volume and get it down to a much more manageable volume. So that was the first thing that we did. And I got to tell you, when you do this work, contamination is always the monster that you're fighting. In fact, we have a hot pink clean room suit. I call it the bunny suit that we wear in the lab, and we have to try to remove plastics everywhere we can. But we're always fighting contamination. And even the reagents that we use, we have to pre-clean them. So we spend hours and hours and days actually just cleaning reagents. So anyhow, that's the first part of the puzzle of being able to analyze nanoplastics. And the second part is the instrumentation. So we use a technique called pyrolysis gas chromatography, mass spectroscopy. And basically all we're doing is the front end is much like a toaster oven. You just heat the sample up instantaneously to a very high temperature. And when you do that, polymers will break down in a very predictable manner. And then we can separate them on a gas chromatograph into their individual components, and then we have a mass spec in the end that can identify them and quantify them. So that's the kind of the technique that and I think that this has the most promise when we do nanoplastic analysis, because I think it's one of the only techniques that will be sensitive and selective enough to achieve this. And this is just to show you what this data looks like. The x-axis is a retention time, so this is when it elutes off the column, and then the y-axis is the intensity, or how concentrated the material is. And this is just showing you a standard. And the point here is all these different plastics, we've identified all these different markers for each type of plastic. In fact, I used to have this chart on my wall with little pieces of string. People thought I was nuts, like I was trying to solve some crime case in my office, but we've identified what the major markers are for each type of plastic, and this is how we can analyze these things down to such a small size. Now, this is actually a ASTM method now, too. This is a standard method for analyzing micro and nanoplastics. So this is what our workflow looks like now. This is how we can actually separate samples into microplastics and nanoplastics. It's extremely labor intensive, and there's a very high cost for every sample. I usually tell people it costs me about $1,000 per sample to do that. So if you thought PFAS was expensive, try this. This is even more. So, but wait, people pay us to do it. So, but anyhow, this is just an example of when we first developed this. What we decided to do is apply it to drinking water samples. So we did a study of looking at a drinking water plant, or actually 2 of them. They were drawing from a surface water, so I'm sorry I didn't do a groundwater study at that time. But we figured surface water would be best because that we expect to find high levels of plastics. And for starters, I'll say that the microplastics, the drinking water facilities did a great job at removing microplastics. About 99.5% of all plastics, microplastics that went in their systems were removed. But we did see nanoplastics. So the bar graph on the left, the blue one is just showing you that was polyethylene that was detected. And you can see the 2 bars on the left, those are the input to the drinking water plant, so that was the surface water that was sampled. And then the next 2 over from them was showing you the output of the drinking water plant. So you can see that it's knocking down the nanoparticles of polyethylene. And same thing with the second, the dotted line is to separate the 2 sites. There was drinking water plant 1 and drinking water plant 2. I can't tell you which plants they were. But anyways, the point is that they're knocking them down. But it makes sense, too, that polyethylene would be the most abundant plastic that we saw in these systems because that's the most abundant plastic that's produced. It's polyethylene. But we did see some other ones as well. It was kind of interesting. Polystyrene seemed to be pretty prevalent in these samples. And again, we saw the drinking water plants were still knocking some of it down. But then we saw some other interesting things like polyamides, like nylon. We saw it actually removed it, all of the nanoplastics for that one. And then polymethyl methacrylate we saw also get removed dramatically by the drinking water treatment plant. And methyl methacrylate is really important because we believe that a lot of these microplastics, or the vast majority of microplastics we find in the environment, we believe has to do with road wear and tire wear particles and road paint. So methyl methacrylates is typically the methacrylates are the paints that they use on road paints. In fact, I spent the summer going around and cutting pieces of road paint off of the roads and analyzing them. So it's definitely, it could be a major contributor to a lot of the environmental microplastics is the methacrylates. But anyhow, yeah, so that's just what I have for today, and I'll be happy to field any questions, and thank you for your attention. Plastic.
You got over 10 minutes.
Oh, boy.
Plenty of time.
What's that?
You got more than 10 minutes, so plenty of time for questions.
Oh, you don't tell them that.
11 minutes of questions.
What challenges exist in conducting studies on them? Oh boy, on the microplastics and health. Yeah, polymers are a lot more, I don't know who asked this, anonymous, okay. Polymers are a lot more different than a dissolved chemical because you not only have the materials, but you also have the particle sizes. So when you do toxicological studies, you're gonna have to look at them at all different particle sizes. And the other thing too is we have so many copolymers. I mean, plastics are incredibly complex. And the other thing too is that most of the studies will be done on pure plastics. Remember, I told you it's more than just plastic. If you don't get anything out of my talk, size matters, and it's more than just plastic. That when we need to think about all the additives that we used in plastic. So that, I think, is the challenges in conducting that. Okay, thank you. Get rid of that one. What's next? Do you have any shareable knowledge regarding the use of bacteria to consume and dispose of? Yeah, you know, I was at a talk the other day where someone was isolating microbes from landfills. And they were trying to find ways to decompose these things better. So yeah, there's a lot of that work going on. But I'm a little scared of it. Because plastic is, you kind of want plastic in some places, right? Like if you put a valve in my heart, I want it to be the best darn material, and I don't want a bacteria to get a taste for it. So I mean, I think you have to be a little careful there. In fact, I knew a guy one time, he told me that they're gonna develop some bacteria and just dump it in the ocean, and it's gonna solve all the plastic problem. But then there's a lot of stuff in the ocean, that plastic that we want, like boats, right, and cable lines. So I don't know. Sorry, I digress. Can we, am I okay on that one?
That's true.
Okay. Can we get rid of that question?
I have no control over that.
Oh, who's got control over that? Who do I blame?
Hideyuki has control of that.
Okay. So I know who to blame. Oh, this one actually gave her name, Amy. Okay, thanks, Amy. Let's see. Sometimes in my outreach to private well users about water testing, I worry that many of them are just reaching towards bottled water. Yeah. Bottled water is terrible, isn't it? Rather than following through with the complexity of treatment, what messaging would be effective to encourage water treatment over bottled water? Price. You want to get somebody's attention, tell them the cost. And I've actually done this with a lot of my neighbors. That was one thing for sure. That'll get everyone's attention. I think I got that one. Katie, get that one out of there. Are you worried about microplastics in beer? Yeah. Some things are worth dying for. But yeah, have you guys seen the one about PFAS in beer? And they found that breweries located their drinking water supplies that had PFAS hits also had PFAS in their beer. So we're going to ruin, us emerging contaminants guys are going to ruin everything. But no, yeah, thank you. Can you please talk about BPA and other main substances that may be noted by manufacturers like BPA free? Can you talk about BPA? Yeah, I think BPA is going to do a pretty good job of phased out. And that's because, again, communication, all you communication people out there, you're so important. You're actually, you're the number one way to fix a lot of these problems. And I think that they did a great job of getting education out there about BPA. And the reason why BPA also works so well is because of labeling, right? You see all that BPA-free labeling. I think that's making people move away from those materials. So I think labeling, in fact, that's a big solution to PFAS, in my opinion, is that if we can get materials, because I think people will move away from PFAS materials if they know which materials have PFAS in them. So give people the tools to do that. What was that? Okay, I'm sorry. Nanoplastics in sunscreen. Yeah, I wouldn't be surprised if they're there. I mean, nanoplastics are pretty much everywhere. But I mean, yeah, I mean, they could potentially cross the skin barrier. Yeah, I don't, does anybody know, who asked me this question? Anonymous. Yeah, this is the one, you can buy me a beer and we could talk about this one because, number one, I got to give it some more thought, but I'd like to know where what nanoplastics that they know that they're there and all that stuff. That one would be better for a longer discussion. Is the increased occurrence of smaller particles for future potential health concerns due to surface area volume ratio and a greater ability of contaminants to absorb these particles? Did you investigate this at all? I have spent about a year studying how contaminants absorb to microplastics. And I came to a conclusion that something would have to eat an ungodly amount of microplastics to really make a difference. I think when it comes to the contaminants that are absorbed by microplastics, that you should be more concerned about the things that you eat, the air that you breathe, the water that you drink, than the exposure from the plastics. So I stopped doing that work. I figured, that's where I go. So I did investigate that. But I still think in terms of plastic, I think additives are really the things that we should be worried about, especially tires. I mean, tires are incredibly complex materials. They're actually more than 10% additives by weight. And every single tire wear company's got its own special sauce. So when you want to talk about, and like I said, tire wear is a major, major source of these things in the environment. Is there a standard for lab equipment that evaluates them for shedding during analysis? There's not a standard one. Actually, you know, I did partake last year in building one that was, we wanted to look at, here we go, I'm going to ruin face masks. Somebody was showing a picture of a face mask earlier, but we were looking at how these N95 masks shed microplastics, and we actually built this device to simulate breathing, and in the first like 10 seconds that I put a mask on, we were filming plastic particles coming off them. So yes, we can actually build these kind of pieces of equipment, but there's no standard lab equipment for that. We have to build them on the fly, which is the fun part. All right, can you analyze, oh boy, yeah, we're working on that one. In fact, I'm sorry, I should repeat the question. Can you analyze human tissue matrices for microplastics and nanoplastics? And the answer is kind of, but there's a lot of, it's kind of like the Wild West right now. In fact, you might have heard about that study in the brain tissue, majorly flawed. You couldn't, I mean, there's major flaws in that. And it was, a lot of what they were seeing was just lipids that were looking like plastics. So there's a lot of work to be done on human tissues. But right now, NIH has got a call for proposals. They got a program called STOMP. It's quite ambitious. In fact, I think it's a little nuts. But they want to develop a sensor that can detect micro and nanoplastics that will give you results in 15 minutes, and it's for less than $50. So yeah, we'll see. I'm very skeptical we're going to see it that fast, but we'll see. But we applied. So, but anyhow, is PFAS absorbed plastic, can they be reduced? Yeah, I get that question all the time. But there's better materials for reducing PFAS. Carbon. Carbon does a really good job at removing PFAS and ion exchange resins and all that stuff. So I would say stick with carbon. Because the other thing too is if you're going to use PFAS to absorb plastics, then we got to worry about nanoplastics getting in the water, right? We got to worry about plastics. And I think carbon and there's other absorbents that would be better than plastic in my opinion. I get that one all the time. Testing for microplastics. Sorry, I had to get some more microplastics. You know, it depends on where they're getting their water. Steve and I have talked about this. It has a lot to do with the source water, right? Deep aquifers, you wouldn't expect to get a lot of plastic all the way down into a deep aquifer. But if you're in a karst system, yes, you do have the potential. I've even seen, you know, I talked to my colleague that was going out collecting the samples for the karst, and he said people were using karst, that the sinkholes as garbage disposal systems. They would put their bag of garbage there, and it would disappear the next day. Well, guess where it was going? Into their karst groundwater system. So definitely, that depends on your source of your water. But I think there's a lot of other things for private homeowners or private well owners that they should be worried about instead of microplastics. Things like nitrates that we're hearing about and lead. I really think that's the priority there.
Last question.
This is the last question? Okay. Ooh, it's a long one. Are biofilms occurring on nanoplastics? Well, probably, but they definitely have a lot. Well, that I don't know. I've never looked at the. Take that back. We've looked at some plastics under SEM, and we do see stuff growing on it. So yes. Or plastic particles small enough to cross the blood-brain barrier. Yeah, they can get to the point where they're small enough, I'm pretty sure, to cross it too. And they're also pretty hydrophobic. I got to be careful, though. I do have a doctor here somewhere. He might call me out on, oh, yeah. But I think that a lot of these materials, it depends on which ones, but if some of them are hydrophobic enough, is that right? Hydrophobicity will make yeah, so they could, at the right size and the right conditions, they can definitely get into the blood-brain barrier. Are there any species found to be associated with these microplastic biofilms? Yes, there are lots of different ones. Sometimes we find on microplastics some of the halophiles, the types of bacteria that like to metabolize organic halides, so like for PVC and stuff. So we definitely see a shift in the diversity of microbes associated with these plastics. Hopefully that answered your question, Patrick. If not, then you can buy me a beer and we could talk about it. Yeah. Well, yeah. That's it? We're good?
That's it. Thank you.